Hello, everyone. Good morning. Good morning, Dr. Wang and Dr. Larkin. My name is Sanil Nadar. I'll be continuing with the presentation what Andrea completed previously. The second half of this study I'll be going over. Since I'm recording this video and then going over the couple of graphs and the study results, I'm using a script, so just to balance out the time so there won't be any gap in between the studies. So um, Dr. Wong, help us out to understand a little bit more better when we have questions and concerns in between, and I'll go back and watch this presentation again when we have the Zoom recording. Thank you very much for this opportunity. So first, I'd like to go over just an overview of the study. I'm sure Andrea might have explained it definitely very well, the background and why the study was done and the basic background. So um, the study is around a couple of basic ideas. The first part is just a characterization of helper NLRs as calcium permeable channels. So it identified and characterizes a subset of nucleotide binding leucine rich repeat receptors, which is otherwise called NLR, known as helper NLR, example ADR1 and NRG1, as capable of forming calcium permeable non-selective cation channels in plant cells. So this is a significant advancement in understanding how these proteins contribute to plant immunity. The second study highlight is like mechanism of immune signal transduction. It demonstrates that the activation of helper NLR leads to the oligomerization and localization to plasma membrane puncta, where they facilitate an influx of cytoplasmic calcium ions. This influx is critical for the downstream signaling processes that activate plant defense mechanisms, including cell death pathways. The third one is the sensitivity to calcium channel blocker. So the study also shows the calcium influx and subsequent cell death mediated by the helper NLR are sensitive to calcium channel blockers. So this sensitivity provides a tool for further dissecting the signal pathway and could potentially offer targets for controlling plant immune responses. The fourth is the role of conserved negatively charged N-terminal residues. That's what the study explains at the later part at the end. It identifies the conserved negatively charged N-terminal amino acid residues in the helper NLR are essential for their function as cation permeable channels. So mutations affecting these residues diminish the ability of the NLR to conduct calcium and induced cell death, highlighting the importance of these residues in the protein's channel forming activity. The next is the every research definitely should have a potential applications in some form of our life. So broad implications for plant immunity. These findings have broad implications for understanding the molecular basis of plant immunity by elucidating a direct mechanism through which helper NLR transduce cell death signals through the formation of calcium permeable channels. So this research adds a crucial piece to the puzzle on how plants detect and respond to the pathogenic threats and potential agricultural applications. Understanding the molecular mechanism underlying helper NLR functions could inform strategies for enhancing crop resistance to pathogens. By manipulating the activity of this calcium permeable channels, it might be possible to develop a crop that is more resilient to disease, reducing the need for chemical pesticides and contributing to sustainable agriculture. So those are some of the applications and contributions of the study. So now we're going to switch over to the major results, and Andrea might have explained the first figure. So I'm going to pick up from the second one. So this is not necessarily one of my strongest area, plant immunity. So please, Dr. Wong, help us to understand maybe, when I go over this, and so I'll go back and learn much more when I rewatch this. All right. So the figure two is actually focusing on um, active NRG 1.1, which is D485. V oligomerizes on the plasma membrane and triggers cell death. So this demonstrates the phenotypic effects of expressing different variants of NRG 1.1 protein. And here the model plant that we used is in the study is Nicotiana benthamiana. At 2 days post inoculation, that's what here it's called as DPI. The figure shows here on how mutation in the NRG 1.1 protein affects the ability to induce the cell death and express in plant cells. And you can see that right here. Let me see if I can get the laser pointer. You can see right here, NRG 1.1, D485, VYFP. So the wild type NRG1 protein, when mutated to the autoactive D485V, which is pretty much we call as DV form, is capable of inducing cell death, which is observable by visual inspection as the tissue necrosis or damage at the site of expression. This autoactivation mimics the natural activation that occurs when a plant's immune system detects a pathogen. However, when specific mutations are introduced into the NRG 1.1 DV protein, such as DN16, L131E, and L125E, the ability to induce the cell death is lost because it's mutated. And these mutations are likely crucial for the protein structure that is necessary for the function as an immune receptor and the subsequent activation of the cell death pathways. And these findings suggest that the full-length NRG 1.1 protein undergoes specific structural changes upon activation that are crucial for its function in immune signaling and the induction of the cell death. This is significant because it highlights the importance of the protein's conformation and localization to the plasma membrane in the plant's defense response. Additionally, the phenotypic changes observed at 2 DPI, 2 days after infection, provides a visual confirmation of the biological activity of the NRG 1.1 protein variants and their role in plant immunity. So figure 2b, this right here, presents the result of the blue native page analysis, which is right on the top here, that we are used to determine the oligomerization of NRG 1.1 DV variants in plant cell. The key findings from this figure can be summarized like active NRG 1.1, which is D485V, an autoactive variant of NRG 1.1, formed high molecular weight complexes indicative of oligomerization. In contrast, the wild type NRG 1.1, which is inactive without sensor NLR activation, did not form such complexes. Additionally, NRG 1.1 DV variants with the specific mutations in the catalytic P loop also did not form oligomeric complexes. Interestingly, the L131E mutation, but not the L125E mutation, when introduced in cis with the DV mutation, stopped the oligomer formation of NRG 1.1 DV. These findings suggest that the L134 residue plays a critical role in the oligomerization process, which is essential for NRG 1.1 cell-mediated cell death. So oligomerization is a key step for NLR to the immune response, as it leads to the formation of active signaling complexes that can trigger downstream response, such as the hypersensitive response and cell death. Therefore, the results explained here in 2b provide insights to the molecular mechanisms underlying the activation and function of NLR's plant immunity. Going to the continuation figure and figure 2, it's the further evidence that is showing here in 2c of the study shows the subcellular localization and enrichment of NRG 1.1 protein variants within the plant cells. In the context of full-length NRG 1.1 in this autoactive form, it was found that the protein is enriched in the plasma membrane fraction, which is key aspect of its role in initiating immune response. This contrasts with the wild type and certain mutant forms, NRG 1.1, which were not enriched in the plasma membrane fraction. In particular, the NRG 1.1 DV variant with the L125E mutation showed the substantial reduction in plasma membrane enrichment, indicate that the mutation affects the protein's ability to localize properly within the cell, which is critical for its function in triggering immune response. So these results demonstrate that the proper localization of NRG 1.1 to the plasma membrane is crucial for its function in plant immunity. The enrichment of NRG 1.1 DV in the plasma membrane fraction correlates with its ability to form oligomer and induces cell death, underscoring the importance of specific protein domains in the correct cellular localization and the function of immune receptors in plants. That's the same thing that continuing to the figure 3. So in figure 3 is explaining NRG 1.1 forms ion channels permeable to calcium and not chloride ions. So in figure 3a, right here, you can see that on x-axis we have different proteins here, and y-axis is the pores per micro square meter, number of pores. It shows the presence and distribution of plasma membrane pores in cells expressing different NRG 1.1 variants, such as would be crucial for demonstrating the pore forming capability of NRG 1.1 when it is activated, like DV variants, compared to its inactive or different mutant forms. So that's what you see here. NRG 1.1 D485V could able to cause more membrane pores. That's what you see here in B, C, and D. These arrows indicates that the plasma membrane pores, which is explained here as the darker spots. So figure 3b to D is presenting the scanning electron microscopy image, providing visual evidence of the plasma membrane pores. These figures would visually highlight the difference in plasma membrane pore formation between the cells expressing the active NRG 1.1 variant and those expressing either inactive forms of variant with the specific mutations that affect their function. The comparison could include cells expressing a known pore forming protein as a control, offering a benchmark for the understanding the size and distribution of pores formed by NRG 1.1. And again, the study did not give a clear detail about the relationship between the size and distribution, what size which protein is, what size pore which protein is causing. At least I couldn't find it. Now the figure 3 is continuing here. Now here what you see is they use another pore causing protein called GSDMN L192D to compare with the NRG 1.1 D485 with their results. And pretty much their results, when you compare in this graph, they are more like similar. So, and the same thing that you see here in this this graph right here on x-axis is over time, and they measured the amount of calcium influx. So this involved multiple steps. Okay, so here in the second graph right here, what you notice, and we are using the two variants. One is the NRG 1.1 DVP loop, which is right here as a control, and this one is the mutant, which is NRG 1.1 D485V. So at around after 30 minutes, researchers added 2.5 millimolar of calcium chloride, and then immediately you can see the calcium influx has been a threshold increase. So this whole entire process happens in multiple steps. The first step is the activation of NRG 1.1. And then the oligomerization step, and then the activated NRG 1.1 complex localized to the plasma membrane. That's the localization step. And then the pore formation. So once the once at the plasma membrane, the oligomerized in NRG 1.1 complex are thought to insert the lipid bilayer forming pores. So these pores are non-selective cation channels, which the researchers later they proved that it is not selected channels. It is not ligand binding. It's a non-selected general channel, allowing the passage of calcium ions and possibly other cations. So that's why we see then influx increase right after, immediately after we add the 2.5 millimolar calcium chloride. The formation of these channels is a direct consequence of the physical insertion of the protein complex into the membrane, altering the membrane's permeability. So once the pores are formed, once the calcium will influx in, these pores act as a secondary messenger, and some of the signaling pathways activates the downstream response that can lead to the programmed cell death. So it will not pass to the other cells, and the infection will be limited. The production of, and also another thing that we can talk about, it's not necessarily mentioned here, maybe the production of reactive oxygen species, and as an expression of defense related genes and formation of pores also, help to kill that particular cell, so that the plants can be saved. And and also there should be a regulation. The activity of NRG 1.1 and the pores it forms are tightly regulated to prevent the unnecessarily damage to the cell. So mechanism likely exists to disassemble the NRG 1.1 complexes and close to the pores, restoring the cell homeostasis after the threat has been neutralized. So that's what you see here. And this study, they repeated is in the animal cells as well, which is coming in the next slide. So what do you see here is the same study, so it's the same graph. And what you see here is in HeLa cells, these red dots indicate that the mutated D485V had an influx of calcium ions. So they used calcium ion staining dye. I don't remember which one was it. So that's what you notice here. The others are the different type of controls. All right. So this is again the continuation of the same figure 3. So use of the ion-specific channel blockers. So just to confirm and validate their finding, it is calcium ions are the one that passing through the spores, not the chloride ions. To validate their findings, so they use the non-specific channel blocker. So the channel blocker that inhibit pretty much the calcium channels, such as lanthanium chloride, LaCl3, and gallium trichloride, which is GDCl3. And observing the effect of calcium influx or cell death induced by the NRG 1.1, and this one can provide the evidence for calcium permeability. So these blockers, and that's what you see in this diagram right here, GDCl3, LaCl3, and this is the normal calcium distribution. And here you can see that. So the one which we used, the blocker, the LaCl3 and GDCl3, and there is no calcium movement. Now, this study also mentioned about, they use another type of calcium blocker, which is RR, I forgot the form, ruthenium something. So this one did not give the same result as the other two, so that may require further experiment. And this is the normal, and this is the control. These are the blocker, and this is the third blocker they used, and which did not necessarily give the same effect as this one. All right. Next, this is the last and final slide from figure 3. So here, what you notice here is, oh, they proved it is not sodium chloride, which just to validate and confirm, and it is the calcium. So what you notice here is they added sodium chloride, and you can see there is no difference between the control and the experimental, both of these samples, 485V and the P loop one. So they added around right after 250 or to 300 seconds, they added around 200 millimolar sodium chloride, and there's no influx of these ions. As you can see, it's actually diffuses in the opposite direction. So, they measured. I haven't added that picture here. So there was in the supplementary figure there was a blue cell counting picture which was given there as well. Sorry about that. You can check that. So that's what this graph is about. So now we're gonna move on to the third part of the uh, results, which is figure 4. In this part of the study, so they explains negatively charged residues in the conserved RNL motif are required for the calcium 2 plus, calcium influx, and cell death. So, what, in summary, what they are talking about here is they mutated some of the negatively charged residues in the conserved RNL. And this is the part of the supplementary figure that shows some of the important conserved area. They actually selected around 18 different amino acids in the multiple different protein samples, which was given in the supplementary figure, but I just used only this one just to focus on the important area. So they confirmed that these negatively charged amino acid residues are the one that it is required for the calcium influx and cell death. So it investigated the role of conserved negatively charged residues in the first 16 amino acid of NRG 1.1 in relation to the calcium influx. By analyzing 334 plant RNL sequences, it was observed that a motif containing glycine or negatively charged or polar residues separated by the hydrophobic amino acid residues was conserved across the ADR1 clade of RNLs, partially degenerated in the NRG1 clade and further degenerated in CNLs. These negatively charged residues were found to be critical for ion selectivity and permeability in calcium channels. Mutations targeting these residues in NRG 1.1 and ADR1 were introduced, resulting in loss of calcium influx and cell death induction. The study underscores the significance of these conserved residues for calcium influx and cell death regulation mediated by RNL proteins. So here, these red arrows and these orange arrows are the conserved residues, which is related to this mutation. All right, so here in figure 4, we are continuing here. So here we used a couple of other mutants and a double mutant here. So this study in this part of the experiment assessed the calcium influx triggered by specific alleles in plants using intracellular calcium reporter, so which is GCAMP3 in this transgenic NB, which is Nicotiana plant. Expression of either NRG 1.1, DV, or ADR1 led to the increase in the intracellular calcium level 2 to 3 hours after estradiol treatment. So here the estradiol is acting as a kind of inducer to turn on the gene, accompanied by the cell death 16 hours later. Treatment with LACL3 and GDCL3 blocked cell death and reduced calcium influx. Mutations in the conserved N-terminal motif of RNL proteins, particularly in E14Q in NRG 1.1, DV, and D11N in ARD1, significantly decreased the rate of calcium influx and attenuated cell death induction both plants and in HeLa cells. And this is a continued diagram. So here, mutations in the conserved N-terminal motif or RNL proteins, particularly E14Q in NRG 1.1 DV and D11N in ADR1, significantly decrease the rate of calcium influx and attenuated cell death induction both in plants and HeLa cells. So NRG 1.1 DVDN16 retained oligomerization and PM enrichment, but not calcium influx. DVP loop refers to a combination of channels in the NLR protein that induces the D485V. Auto activating mutation, that additional mutations in the P loop, which is a phosphate binding loop essential for the protein's ATPase activity. That's what you see in the first diagrams actually. The DV mutation is known to cause autoactivation of the protein, while the P-loop mutations may affect its nucleotide binding and hydrolysis functions, which are crucial for the protein's ability to signal and initiate immune response. So here in this is the HeLa cells. They replicate the same thing for the cross species verification. In animal cells, they respond. So in both plant system and HeLa cells, that's what you see here. Alterations in the conserved N-terminal motif or RNL proteins, particularly E14Q in NRG 1.1 DV and D11N in ADR1, notably diminished the pace of calcium influx. And furthermore, this modification results in the decrease in the initiation of the cell death. I'm going to quickly rush through because it's 27 minutes already. So this one is a separate confirmatory assay. They measured the voltage, calcium ion voltage inside the cell or across the membrane using something called patch clamp technique. So that's what you see here in both this diagram. The graph describes the analysis of the electrophysiological properties of NRG 1.1 and its variant, which is DVD3N E14Q in human embryonic kidney cells. So they used kidney cells to do this experiment. All right, using the patch clamp technique. After confirming that NRG 1.1 DV induces calcium influx in transfected HEK293 cells, similar to HeLa cells, whole cell voltage clamp currents were recorded with the voltage ramp. Robust current were observed in cells expressing NRG 1.1 DV with the specific ion solutions in the pipette and the bath. These currents were abolished when non-selective cation channels were substituted, indicating that NRG 1.1 DV forms a non-selective cation channels mediating calcium influx rather than chloride influx. The NRG 1.1 D3N E14Q double mutant eliminated calcium currents, suggesting that D3 E14 act as a selectively filter. The patch clamp technique is a laboratory method used to study the electrical activity of the individual cells. So I'm not going to explain that because of the time. All right, so this one is a confirmatory assay, again, to validate the finding. Substituting the magnesium with the magnesium for calcium resulted in similar permeability, confirming that NRG 1.1 functions as a non-selective cation permeable channel. That's what you see here. They use the TEA as a control, and then here's the calcium channel. So you can see that D485 has a higher influx. And this is the double mutant. And the TEA is a tetraethyl ammonium. Is used to electrophysiological experiments, including those described here in the NLR proteins, and their function as an ionic channels, primarily because its ability to block certain types of channels, that including potassium or pretty much any positive channels, I believe, ions that transport positive charged ions. The last diagram here, this is actually from a supplementary figure. supplementary figures. So they repeated the same experiment in a new model. So what they did here is the representative time course measurement of intracellular um, calcium ions. That's what it is measured here. So RPS4, which is the resistance to Pseudomonas syringae 4, is a plant immune receptor protein belonging to the class of TNL proteins. It plays a crucial role in the defense responsible of plants against certain pathogens, particularly the bacterial pathogen, which is Pseudomonas syringae. RPS4 is involved in recognizing specific pathogen effectors and triggering immune response, including the activation of defense pathways and cell death in infected plant cells. It is one of the well-studied example of TNL immune receptors in plants. All right, so now coming last part, the discussion. So major findings in the study, or some of the key ideas that in the study is calcium signaling plays a crucial role in plant immunity. Constitutive cytoplasmic calcium influx lead to the defense activation and cell death. NRG 1.1 protein structure assembles the pore-forming domain of MLKL and forms oligomers on the cell membrane. That explains in the figure 1 different molecular structures there. NRG 1.1 drives cytoplasmic calcium influx in both plant and human cell, both HeLa cells and the kidney cells. TNL immune receptors require RNLs from the ADR1 and NRG1 subfamilies for activation, suggesting a role of calcium influx in initiating cell death and immune response. Activation of TNL RPS4 is associated with the calcium influx. Cyclic nucleotide gated channels may sustain calcium influx initiated by RNLs. CNLs likely function similar to RNLs by triggering calcium influx. Negative charge rings in proteins like NRG 1.1 and ADR1 act as a cation selective filters, facilitating rapid calcium influx and initiating cell death and defense signaling upon pathogen recognition. These findings provide insights into how TNLs and CNLs control the cell death in response to the pathogen recognition in plants. So this is just a quick overview of the study. I know it is not necessarily a perfect way of doing it, but thank you very much for your attention. Thank you.